Welcome back, students. So here it is your Arka sir. So I have come with another episode on biology. So today I will discuss a very important topic of biology, that is the blood, human blood. Especially we will discuss in this video. Totally I cannot complete in a single video. Some more videos I have to upload. If you can follow the video from first to last without skipping, I can guarantee you that your concept will be clear. We will basically discuss what is blood, what is the function of blood, why it is known as the liquid connective tissues, what are the components, what are the diseases may occur if there is a disbalance of the component, its presence in greater or lesser amount, what are their functions, what are the components present, what are the functions of each component, everything in details. So without further delay, let us start. And before I start, if you don't know the channel, I am telling the name of the channel is Physics Scholar, and subscribe the channel, share the channel. So I think someone should get benefit from it. So that is the motto and objective of this channel. So blood, what is the blood? Blood, perhaps the name we all heard from our childhood. We can see, or we see also sometimes if there is injury, blood is coming out. Okay, so we see that. Basically, blood is, what is the function of blood? Actually, the main function of blood is whatever in the digestion process I have discussed, the food are being digested. Why? To convert it into the simple form. So the protein will convert it into amino acid, the carbohydrate will will be converted into glucose, fructose and galactose, that is monosaccharide, and the fat will be converted into fatty acid and glycerol after digestion. So what will happen? That needs to be absorbed from our small intestine and needs to be sent to the cell. So this part, the blood comes. Blood will absorb the food, will take the food, and it will be carried to each and every tissues or cells of our body. And also during the respiration process, some toxic materials will be generated in our cell. So that will come out and mixed with the blood. So that is, and it will then come out in form of urine or sweating, okay, and other processes are there. So that will come out. Another thing is that the blood actually, we are taking breath, and the oxygen will be absorbed by the blood, and that will be supplied to each and every tissue, as well as the carbon dioxide will mix with the blood, and that will again come to the lung in the alveolus. That part, the exchange of carbon dioxide will be there and oxygen taking will be there. So we are breathing out. So that's why we are giving out the oxygen from our body, the carbon dioxide from our body. Okay. So that is the main function of blood. Now what is the blood? Why the color of blood is red? What is the size of each cell, each component? So that we will discuss in details. So first, what is blood? The definition. The definition of blood, we can say, what will be the definition, or in a single word, one or two lines we can define blood. So it is a liquid connective tissue which make a coordination or connection between all the parts of our body, and it is slightly basic in nature. Okay, it is slightly basic in nature. So that is what? That is blood. So blood is a liquid connective tissues In case of the closed circular system, what is closed and open circular system, I will discuss. Okay, so it will be carried through the blood vessels to heart in case of the closed circular system, and in case of open, it will lie within the heart, blood vessels and lacula. Okay, so it will make a communication or coordination, okay, between each and every tissues. It will carry the required things and remove out the excretory products, and also carry some hormones and other things, other required things, and it is slightly basic in nature. Okay, so pH is slightly basic in nature. So that is known as blood. Now our next point is why the blood is treated as liquid connective tissues. Why the blood is treated as liquid connective tissue. Okay, why the blood is treated as liquid connective tissue? So the first point will be it generates from the mesoderm of embryo. So the blood, from where the blood is generated? The blood is generated from where it is actually producing. So when the embryo is formed, okay, what is embryo, what is zygote, okay, what is reproductive cells, that I will discuss in the process of reproduction, in the chapter of reproduction. So embryo, it generates from the mesoderm of embryo. The blood actually starts generating from the mesoderm. Okay, number just like the other tissues. Okay, so number B, the percentage of plasma, it is how much? 55%. Okay, is more than solid particles. Okay, or the other blood cells. The percentage of plasma is 55%, and the blood cells means RBC, WBC and platelets, those are 45%. So the percentage of plasma is more, okay, than the other. Clear? Next, so it carries the required materials to other tissues and remove excretory products. Okay, so these are the reasons why the blood is considered as a liquid connective tissues. First one, it generates from the mesoderm of the embryo. The percentage of plasma is more than the blood cells. It carries the required materials to each and every tissues of our body, as well as remove the excretory product from the cell. So this is why it is known as the liquid connective tissues. So now next topic is the color of blood. Next topic is the color of blood. So color of blood. So human, mammals, you see the color of blood is red. Why Why the color of blood is red? Because hemoglobin is there in the blood. Hemoglobin is made of the heme plus globin. That is nothing but iron is present in heme. Okay, so that's why for the hemoglobin the color of the blood is red. Next one, for crabs, prawn, etc., the color of blood is blue. Why the color of blood is blue? Because instead of hemoglobin, hemocyanin, okay, hemocyanin is present. Hemocyanin is present. Okay, basically copper is present in hemocyanin. That's why the blood of the prawns, crabs, etc., the color of blood is blue. Some insects are there, okay, just like cockroaches and other insects. The blood don't have any color because nothing is present. No hemoglobin, hemocyanin are present. So that's why their color of their blood is no color, actually colorless. Clear? So now let us discuss the next topic. Buffer of blood. First we have to understand what is buffer. Buffer means the component which are present in the blood and which is maintaining the pH balance of the blood. That means it will maintain a constant pH balance of the blood. The value of pH will not increase or decrease. So those components are known as buffer. Okay. So it helps to maintain, to maintain pH balance of the blood. So what are the buffers present in the blood? Bicarbonate buffer, hemoglobin buffer, these are the buffer present in the blood. That's why the pH of the blood will remain constant. Okay, it will help the blood to maintain the pH level. That means a slightly acidic, it has a particular pH level, so it helps to maintain that. So next is the amount of blood. Blood present in human body. Okay, the amount of blood present in human body. In case of the male, healthy male, the amount of blood is 5 liters, and for healthy adult male, and for female it is 4.5 liter. The amount of blood is 4.5 liter for healthy adult female. So that is the amount of blood present in human body. The total amount of blood present in adult male is 5 liter and adult female is 4.5 liters. Next topic. So our next, these all topics are very important for the exam as well as for NEET exam. Those who are preparing for the NEET, some basic things are very important to understand. Now hematocrit value. Hematocrit value. What is hematocrit value? Okay, hematocrit value basically the rate of rate of RBC sedimented okay, the rate of RBC sedimented. Nowadays it is not RBC, it is all blood corpuscles okay, that means also WBC plus platelets. So one instrument is there, the name of the instrument is hematocrit okay. So in the hematocrit, if we have some blood inside the hematocrit and if the blood is circulated 3000 times per minute for 4 to 5 minutes, then what will happen? For 4 to 5 minutes, then what will happen? The plasma will come up, the plasma will go upwards and the blood corpuscles will be saturated okay at the bottom of the machine. So this is known as, this will be 45%. 45% will be the blood corpuscles. So this value is known as the hematocrit value. This is also known as the PVC or packed PCV, packed cell volume. This is also known as PCV or packed cell volume. Clear? So hematocrit value, the rate of RBC sedimentation. Okay. Now instead of RBC, we are considering, now the doctors and the scientists are considering the all blood corpuscles, that means including WBC and platelets. So actually how it is done, hematocrit is a machine. So if we circulate the blood inside the machine 3000 times per minute for 4 to 5 minutes, we continue this process. Then if we just keep it for some time, then what will happen? The plasma will go up, the plasma and the blood corpuscles will be separated from each other. The plasma will come up okay, and all the blood corpuscles will be saturated at the bottom layer of the machine. So the percentage of it will be 45% because 55% are the blood plasma. So this value is known as hematocrit value or PCV, packed cell volume. That means all the cells, corpuscles are packed together. That is known as packed cell volume. Coming up to the next topic. Next is erythrocyte sedimentation value. Erythrocyte sedimentation value. What is erythrocyte sedimentation value? Okay. So if we mixed oxalate salt with blood. Oxalate salt, why? Because it is anticoagulant. That means it will stop blood to be coagulated. Clear? So if we have a uniform test tube, the radius of each portion is same and the blood is kept, blood mixed with oxalate is kept here for some time. Then what will happen? Then the RBC will be sedimented at the bottom and the plasma will be at the top. Okay. So this value is known as erythrocyte sedimentation value. For male it is 0 to 5 mm per hour. For female it is 0 to 7 mm per hour. So this is the erythrocyte sedimentation value, and also it is known as also ESR. So the blood test, many times the doctors prescribe to do ESR. So that is erythrocyte sedimentation value, okay, or rate, erythrocyte sedimentation rate. Okay. So this is actually useful to identify the nature of some diseases. Okay, so ESR or erythrocyte sedimentation rate. So next, let us move to the next topic. Now next topic is blood components. Blood components. Okay. So mainly two components are there in blood. First one is plasma, and next one is blood corpuscles. Blood corpuscles basically are three types: RBC, WBC and platelets. So there are some classification of WBC, function of RBC as there, hemoglobin is there in RBC. Okay, so these are basically the two components of blood. So today in this video we will discuss about the plasma and RBC, and the next video we will discuss in details WBC and platelets. So the blood basically have two parts. One is the blood plasma, that means the liquid part of the blood, and the blood corpuscles. Okay, so blood corpuscles have three parts: RBC, WBC and platelets. Clear? So now today we will discuss in the video plasma and RBC. Okay, so let us move to the next topic. Okay, so now we will discuss about the constituents of plasma. That means which are the components basically are there in plasma. So first one is water. So the percentage of water in plasma, it's a liquid, that's the liquid part of the blood, that is the percentage of water is 90 to 92%. Mineral salt. So some mineral salt are basically there in the plasma, which are there: sodium, potassium, calcium, magnesium. Okay, so phosphorus. So these are the mineral salt of the plasma. Now nutrients. Nutrients means those which are we absorbed, okay, after digestion. So what? The protein will be amino acid, the carbohydrate will be glucose, fructose, galactose, and the fat will be fatty acid, glycerol. So these. So those which are which we generate after digestion. So glucose, fatty acid, amino acid, okay, fructose, galactose. Okay, so these are the nutrients. Now plasma protein, there are some plasma proteins are there: albumin, globulin, fibrinogen, prothrombin. So fibrinogen, prothrombin, these actually helps blood coagulation. So when we will discuss about the blood coagulation, we will discuss about the functions of the fibrinogen. There is also prothrombin. Okay, so albumin, globulin, these are the plasma protein. Dissolved gas, what I told that basically hemoglobin basically carries the oxygen to the cell and remove the carbon dioxide to the cell. But small amount of oxygen and carbon dioxide are also carried by the plasma, okay, in the dissolved form. Okay, the oxygen will be dissolved. A small amount of oxygen will be carried by plasma to the different cells and from the tissues, and from there it will also dissolve some amount of small amount of carbon dioxide and it will be carried. So some amount of oxygen and carbon dioxide can also be seen in the plasma. And the other components, what are the other components? Other components is heparin. Heparin is the anticoagulant. That means due to the presence of the heparin, the blood does not coagulate or cannot coagulate in our blood vessels. Okay, so it is a anticoagulant substance that is present in plasma. Heparin, some hormones are there. Okay, some hormones are there. Clear? So these are actually the other component that are also present at plasma. So basically, so these are the main component which are constituents of the plasma. So first the water, the main component of the plasma. The mineral salts are there, dissolved at plasma. The nutrients what we absorbed from our stomach from our small intestine after digestion. Then plasma protein, albumin, globulin, prothrombin, fibrinogen. Basically fibrinogen, prothrombin helps in coagulation of blood. Dissolved gas, oxygen and carbon dioxide, a small amount of, mostly are carried by the hemoglobin of RBC. And the other component, heparin, hormones, etc., the other component which are present at blood plasma. Okay, so these are the main component of blood plasma. Very often it comes like this in the examination: what are the nutrients present in, write some nutrients which are present in blood plasma. So what you have to write whatever is absorbed from our small intestine: glucose, fructose, galactose, fatty acid, glycerol, amino acid, plasma protein, albumin, globulin, prothrombin, fibrinogen, okay, and other component. What are the other component? Heparin, okay, hormones. These are the other components which are present in plasma. So coming up to next topic is function of plasma. By the component, we already discussed what are the components present in plasma. We can easily say the function. So first, transportation of nutrients. What are the nutrients present? Nutrients present: glucose, fructose, galactose, then amino acid, fatty acid, glycerol. So it will be absorbed from small intestine and mixed with the blood, and it will be carried to the different tissues of our body, and it will be absorbed from there. So that carried will be done by the plasma. Next one, heparin is there. Heparin is there. It is anticoagulant. So heparin is present at plasma. Anticoagulant, thus it prevents the blood clotting inside our body. It prevents blood clotting, blood coagulation inside blood vessels. Next, plasma protein. Plasma protein what? Prothrombin, fibrinogen, these are the plasma protein. These plasma protein basically help in coagulation of blood when there is any injury. Okay, so fibrinogen, prothrombin, this actually helps blood coagulation for any injury. So this helps blood coagulation, plasma protein. The transportation of nutrients, then heparin is there present in the plasma, which protect the blood to be coagulated inside the blood vessels. Okay, and plasma proteins, prothrombin, fibrinogen, etc. This helps the blood coagulation for any injury. Okay, so next, temperature maintaining body temperature. So if body temperature increases, then what will happen? The blood starts flowing more. Okay, so it will absorb some temperatures from the body. So that's why it will maintaining the body temperature. Okay, next, maintaining the body temperature. So the due to the buffer action, the pH of blood is maintained. So bicarbonate buffer, hemoglobin buffer, these all are present in plasma. Okay, so due to the buffer action, the pH of blood is maintained. Okay, basically the pH of human blood is 7.4, slightly basic in nature. The pH of blood is 7.4 generally. Okay, so these are basically the functions of the plasma. Okay, so hope you understood this. If you can write, you can write it also. So these are the functions of plasma, and just before this I discussed about the different components present in plasma, okay, or carried by plasma. So that is, so let us move to the next part. Now RBC. Okay, so what is RBC first? RBC means the red blood corpuscles. corpuscles, okay, which have hemoglobin and carries O2 to each tissues of our body and remove CO2 from each tissues of our body. So that is the main function of RBC. So hemoglobin is there, that's why it is red in color, and it carries O2, oxygen, to each and every part of our body, to each and every cells or tissues of our body, and also remove the carbon dioxide which is generated during the respiration, that will come out from the cells or tissues and mixed with the blood, again the RBC. Most of it, a small portions are carried by plasma, but most of it are carried by RBC. That is the main function of RBC. So what is the site of RBC? It is biconcave, okay, biconcave. So it is the site of the RBC is biconcave. The site of the RBC is biconcave, okay. So that's why it has more surface area, so that the oxygen, it can get contact with the oxygen, okay. So more surface area is there for this particular biconcave shape. It has more surface area, so it can come contact with the oxygen or carbon dioxide, so that more amount of oxygen can attach with it, okay, and it can be carried. Clear? And the RBC don't have any nucleus. Why? And in the place of nucleus, the hemoglobin can take place, that space. So more amount of hemoglobin, so nucleus is not there in RBC. So in place of nucleus, is not there. That's why the more space are vacant, so hemoglobin can take place. And the size of the RBC is biconcave. So that what is the benefit of that size? More amount of oxygen can get contact, or carbon dioxide can get contact with the surface of RBC. And also if the density of the blood decreases, okay, that means it is dilute. If blood is diluted, the density decreases, or density decreases, then what happened? Then also the nucleus for its shape, the RBC for its shape will not break, but it will make a shape of a ping pong balls, oval, round in shape, okay. The RBC will be round in shape, just like a ping pong ball, and this process is known as phagocytosis. Okay. So if the blood will be diluted, that means density of the blood will be decreased, then that means it will be light. In that case also it will not be broken, okay. The RBC for its special shape, it will not be broken, and it will be in the round shape, just like a ping pong balls, okay. And this process is known as phagocytosis. Clear? So this is the site of RBC and the definition of RBC. So let us move to the next topic. And development of RBC. So first, at the time of embryo, next before one month of birth, and next after birth. Okay, before 1 month of the birth and after birth. Okay, so at the time of embryo, when the embryo will be formed, then the RBC will be generated from vasculosa. Okay, before 1 month it will from liver and spleen. And after birth red bone marrow. Okay, so at the time of embryo, when the embryo will be formed, then the RBC will be start generating from vasculosa region, and before 1 month of the birth liver and spleen, and after the birth red bone marrow. Okay, so these are the part of human body from where the RBC will be generated. Clear? So you can write it in your copy also. So now let us write the function of RBC. Write the function of RBC. So first function is it carries O2 to each and every tissues and remove CO2 from each tissues or of our body. Number 2, the pH balance, it will what? It will maintain. How? Because hemoglobin buffer is there, that's why it will maintain the pH balance. How it will carry the O2 and CO2? So when I will discuss the hemoglobin part, I will discuss in detail. So O2 will react with the hemoglobin and it will form the carboxyhemoglobin compound. So that will be carried up to the cell, and then it will be broken and oxygen will be released and enter in the cell, and the carbon dioxide will come and carboxyhemoglobin it will be formed, and that compound will be flow through blood. Okay, and when it will come to the lungs, then that compound will be broken down and the carbon dioxide will be released. So that part I will discuss in details when I will discuss about hemoglobin. So pH balance it maintains. Okay, so of the blood. So these are the basically the main function of RBC or red blood corpuscles. Okay, so next move to the next topic. Okay. So now we will discuss about the one term is very common before we end the RBC, that is hemolysis. What is hemolysis? If we immerse the RBC inside a liquid having less dense liquid, then what will happen? The water will enter inside the RBC. Okay, so if we immerse the RBC in a less dense liquid, then water will enter inside RBC. So what it will be? What will happen? The volume of the RBC will increase. Volume will increase, and what will happen? The hemoglobin will come out from RBC. Okay, so the volume of the RBC will increase, it will be swollen. Clear? Because the water will enter, but the hemoglobin will come out from the RBC. So this is known as hemolysis. This is known as hemolysis. So the coming out of hemoglobin from RBC, if it is immersed inside a liquid, dense liquid, okay, so then this is known as hemolysis. Clear? What is hemolysis? Very common term for the exams. Okay, what is hemolysis? So coming to the next. Now different type of anemia we will discuss. Okay, anemia means the RBC will destroy. That means the scarcity of RBC will be there in the body. So first one is sickle cell anemia. Sickle cell anemia. Okay, sickle cell anemia, that means it caused due to the destroying at high rate, destroying of RBC at high rate. The size of RBC will be like sickle. Okay, so what is alpha chain, what is beta chain, that we will discuss later on when I will discuss about the hemoglobin. So basically it occurs due to the defect of beta chain of hemoglobin. Okay, so this is what sickle cell anemia. So sickle cell anemia basically it's caused due to the high rate of destroying of the RBC. What happened actually, the size of the RBC will be just like sickle cell. Okay, just like sickle cell, like this. Okay, just like the sickle cell. Okay, and what happened actually, it happens due to the defect of beta chain of hemoglobin. What is alpha chain, beta chain of hemoglobin? I will discuss when I will teach the hemoglobin part. Clear? So now moving to the next topic. Now the next topic is sickle cell anemia completed. Normocyte anemia. Normocyte anemia. Basically normocyte anemia occurs due to the heavy bleeding and loss of RBC from the body due to heavy bleeding and loss of RBC from body. Okay, that is normocyte anemia. So the RBC will also come out from the body okay, with blood. So due to the heavy bleeding and loss of RBC from the body, the normocyte anemia occurs. Next, so what is normocyte anemia? Due to the heavy bleeding. And next anemia is parietal anemia. Okay, so basically it occurs due to the scarcity of vitamin B12 and the inside our stomach, the peritoneal cells are destroyed. So this type of anemia occurs due to the scarcity of vitamin B12. Clear? So parietal anemia. So moving up to the next topic. So this parietal is also known as pernicious anemia. Next is thalassemia. Okay. So thalassemia, basically it's a genetical disease, thalassemia. So thalassemia, what happened actually, the synthesis of hemoglobin will not be there. So it's a genetical disease. So this is what thalassemia. Okay. So thalassemia basically is a genetical disease. So the synthesis of hemoglobin will not be there, and as a absent of hemoglobin, the oxygen will not be carried, as well as the carbon dioxide will not be removed from the cell properly. That's why it's a very dangerous disease. So students, so hope the RBC part as well as the plasma part, and what is blood, what is hematocrit value, what is erythrocyte sedimentation rate. Okay, so you all understand this. So in my next video I will upload the hemoglobin in details. Okay, the alpha chain, beta chain, everything, the structure of hemoglobins, everything, how it carries the blood. Okay, the oxygen and carbon dioxide, and then the main part of the blood is WBC. Okay, the WBC, two parts are there. One is granulocyte and agranulocyte. Okay, in granulocyte, agranulocyte there is neutrophil, basophil and eosinophil, and granulocyte is monocyte and lymphocyte, and also the platelets. Then slowly we will move to the blood vessels, the closed circular system, open circular system, and the human heart and all. So hope you understood this episode. And if you have any doubt on any queries, you can write in my comments. If you want the notes, then also you can ask me for that. I will send it to you, and continue to watch my videos and wait for the next one. It will be uploaded very soon. Thank you.